next presenter is Dr. Yuchen Chan. Dr. Han will discuss intertumoral molecular heterogeneity of non-small cell lung cancer with MET Exon 14 skipping, and this will be a virtual presentation.
Good morning. I'm Yuchen Han, a pathologist from Shanghai Chest Hospital and Shanghai Jiao Tong University. I'm very excited to be here today to share the work of intertumoral molecular heterogeneity of non-small cell lung cancer with MET Exon 14 skipping. Disclosure. Introduction. MET Exon 14 skipping mutation is a rare driver for NSCLCs, which is present about 3% of NSCLCs. NSCLC patients with MET Exon 14 skipping are characterized by a higher frequency in pleomorphic carcinoma and adenosquamous cell carcinoma than in adenocarcinoma. Recently, the approval of MET TKIs, sorafenib, cabozantinib, tabetinib for NSCLCs carrying MET Exon 14 skipping dawn a new era for MET targeted therapy. These drugs displayed encouraging clinical efficiency, yield progression-free survival of 5.4 to 12.2 months in clinical trials. However, it has been reported that 1/3 to half of patients saw inherent resistance to MET TKIs, which indicated biological heterogeneity in NSCLCs driven by MET Exon 14 skipping. This is the flowchart of retyping MET Exon 14 skipping. A total of 10,525 previously untreated NSCLCs from April 2017 to December 2020 at Shanghai Chest Hospital were included in this study. DNA-based target NGS and sequencing analysis were performed, and from those, 133 cases were detected with MET Exon 14 skipping. Then, other samples were subjected to target RNA sequencing using panel consisting of 2,660 oncoimmunology genes. Finally, 126 cases were confirmed with MET Exon 14 skipping positive MET Exon 14 skipping. Single sample gene set enrichment analysis and pathway level unsupervised clustering conducted to identify molecular subtypes in MET Exon 14 skipping positive patients. Subtype-specific pathways, tumor microenvironment, and clinical pathological features were further analyzed. 4 molecular subtypes, including subtype A, B, C, and D, were established. Subtype A, accounting for 33.3%, were characterized by activation of MET signaling, such as upregulation of PTK2, cell mortality, degradation of extracellular matrix at the cell cycle, etc. Subtype B, accounting for 15.1%, was a mixed subtype. It was associated with fatty acid metabolism as well as response to oxidative stress. Subtype C, accounting for 14.3%, was characterized as immune activation phenotype. And subtype D, 37.3%, displayed bypass activation of oncogenic pathway like Notch, MAP, ERP, PI3K, AKT, and NTRK signaling pathway. Immune microenvironment analysis suggested that subtype A was characterized as immune suppressive phenotype with higher infiltration of Treg cells. On the contrary, subtype C displayed higher infiltration of T cells and tumor-associated macrophage, as well as high level of MHC2 signatures. For correlation between subtypes and clinical pathological features, subtype A was significantly associated with more aggressive clinical characteristics. For example, more advanced tumor stage, larger tumor size, and a higher percentage of invasive phenotype morphology, and numerical shorter disease-free survival. 11 patients, including 8 cases of subtype A and 3 cases of subtype D, were treated with MET TKIs. The survival probability analysis showed that these two subtypes have significant differences in the efficiency of MET TKIs. This study disclosed the clinical relevant intertumoral heterogeneity of NSCLCs driven by MET exon 14 skipping. Based on the molecular subtyping, subtype A was more sensitive to MET TKIs, while subtype D was putatively resistant to MET TKIs. Of note, subtype C might be more vulnerable to immunotherapy. Thank you for your attention.